Today, what we want to do is to consider a new type of growth which developed in plants. The type of growth which allowed a plant to go from a twig or a small, narrow cylinder like this to a giant cylinder like this, to make a structure known as a tree, which is the largest living organism and the most massive and oldest living organisms that have ever inhabited the earth. Now, up until the point today, we have talked about primary growth. And we said that primary growth resulted from derivatives from the apical meristem and that these derivatives which formed yielded elongation growth. That is the lengthening of the stem axis. Today what we want to do is to try and understand how this twig, which is of a relatively narrow axis, can increase in girth to become something as massive as this piece of wood or this piece of tree that I'm holding here. In order for us to appreciate this new mechanism which developed, I want to take you back to the point that we left off last time with a cross-section of a stem showing primary growth. So a cross-section of a stem showing primary growth. This will just be a review for you. And these pictures are also in your notes, so that if you want to just watch, they're also in your notes. So we'll make this cross-section. And in the cross-section of this stem, we drew last time, we had a vascular bundle. 4 vascular bundles. And we said that each vascular bundle was divided approximately in half, with the tissue toward the outside of the bundle being the phloem, and the tissue toward the inside of the bundle being the xylem. Now, in order to understand how this stem showing primary growth, which is relatively narrow in diameter, begins to widen or to increase in girth, the first thing we want to do is to focus on one of the vascular bundles. So if we wait for some time, what we find, and we'll just make this cross-section of this eudicot stem again, what we find, and we'll just make one bundle just for our discussion here between the phloem and the xylem, what we find is that cell divisions increase between the primary xylem and primary phloem. That's the first place we see an increase in cell division. And with some more time, if we look at the stem now, so here we'll put the 4 bundles on, just that we dealt with before. So we're saying that cell divisions are increasing here, are increasing at this point between the primary phloem and the primary xylem. At the next stage, what we find is that now cell divisions are spreading to regions between the bundles. So if you could now look at a cross-section of a stem which has this cell division, we would have our bundles again that we had from before, but now we would have a region running through the bundles and all the way around between the bundles. This region or zone of increased cell division activity. So it begins cell divisions between the xylem and the phloem and then spreads between the bundles. Now, if we wait for some more time, what we notice is the following. Quite remarkable. Here's our cross-section of our eudicot stem again. And now what we find is that the primary xylem and the primary phloem, here would be the xylem, and here would be the phloem. The primary xylem and primary phloem are beginning to separate from each other. Now, why is this separation occurring? What is going on to cause the primary xylem and primary phloem to move apart from each other? So if we make our cross-section again of our stem, and we'll just put one bundle here for the sake of it, which is separated, what we find is that new cells formed between the primary xylem are pushing these cells apart from each other, are pushing these cells are moving apart from each other. So we would again, as we drew before, show the primary phloem, the primary xylem. And these have now moved apart from each other because new cell divisions have been produced in the region between the primary phloem and the primary xylem. And if we look very carefully at this stem, what we find is that the zone of new cell production extends the entire circumference of the stem. So what we find is that we have new cell divisions extending the entire circumference of the stem. These are new cell divisions. And that they can be traced to the activities of a new cell dividing layer. So what we can find is that in the middle of these new cell divisions, a new layer of cells which is able to divide, making new cells to the outside and new cells to the inside. This new cell layer is now formed, and it's a complete circle around the stem. This new cell layer is called the vascular cambium. Called the vascular cambium. And it is a meristem because it is giving rise to new cells. But the important point that you should understand is that these new cells which are arising in the plant are not coming from the apical meristem. They are coming from a lateral meristem. A lateral meristem. And because these new cells, I'll write this out for you because this is an important point. Because new cells coming from vascular cambium, I'll just abbreviate that as VC, because these new cells come from the vascular cambium and not the apical meristems, we call or describe these new cells as being secondary. Secondary. A very important concept. So now you're seeing in the plant new cells being produced, but their origins can't be traced to the apical meristems. They can be traced to this lateral meristem, which we're going to talk more about for most of the lecture today. So what are some of the consequences of the operation of the vascular cambium? So the consequences. Of vascular cambium activity. The first one should be obvious to you, but we're going to say it. The primary xylem and the pith are pushed to the center of the stem. So if you look at this picture that we drew up on the top over here, what's happening is that the primary xylem is moving toward the center of the stem, and inside is the pith. So it is being transferred to the center of the stem. The second consequence is that the primary phloem, epidermis, and cortex are being pushed to the outside of the stem. The third consequence of the operation of the vascular cambium is that we make new cells, or new, let's say cells, tissues, to the inside of the vascular cambium. And these new cells are secondary xylem. These new cells are secondary xylem. They are being produced by some other meristem other than the apical meristem. Secondary xylem has a more common name that all of you are familiar with. Secondary xylem is wood. So to the inside, we produce wood as a result of the activities of the vascular cambium. Fourth, to the outside of the vascular cambium, we produce secondary phloem. Secondary phloem. And the common name for secondary phloem that all of you are familiar with is bark. And finally, the last major consequence that I want to discuss with you now is that as a result of producing tissues, namely secondary xylem, to its inside, the vascular cambium has to enlarge in circumference. I'll show you what this means in a moment. So let's look at two cross-sections of two different stems of different diameters. Here is one stem of one diameter and another stem of a much larger diameter. And into both of these stems, I'm going to say that they have a vascular cambium operating, which I'll just indicate as the dashed line. So this is the vascular cambium in both stems. Now let's assume that the stem on the left is the younger stage of the stem on the right. In order for this vascular cambium to remain complete and not broken up into pieces, as it produces new cells to the inside, it's pushing itself out. Everybody get that? It's a three-dimensional sort of view I'm asking you. As it's producing new cells to the inside, it's pushing itself out. And hence, in order for it to remain a complete cylinder, it has to add cells. So the vascular cambium has to add cells to its circumference in order to remain intact. And here's an example that I'd like you to think about. Here is a tube of a relatively small diameter. I'd like you all to watch this because this will help you. A tube of a relatively small diameter. If you could imagine this tube as being the lateral meristem, the vascular cambium, to its outside it's producing secondary phloem, and to its inside it's producing secondary xylem. As it is producing secondary xylem to the inside, it has to continue to expand in circumference in order for it to remain intact, because more new cells are being produced toward the inside. So the vascular cambium is a constantly expanding circumference as it's active, as it is making secondary xylem to the inside. Now, what do we know about the vascular cambium? Well, first of all, if you take a tree like this one over here, the vascular cambium is not simply a circle, but it actually represents a cylinder, like a sleeve, that runs up and down the entire length of the tree. This cylinder or sleeve that covers the entire length of the tree is internal. We can't see it. It is very difficult for us to experimentally manipulate the vascular cambium. Most people now believe that the vascular cambium is a single layer of cells, but we haven't proven that for sure because of its operation on the inside. Indeed, all of the wood that civilization has used since humanity started using wood is a result of the activities of the vascular cambium, the single layer of cells. And yet, amazingly, we do not understand how to manipulate the cambium. We understand very little about its physiology, biochemistry, in order for us to improve the quality of wood, all of which arises from the vascular cambium. So this is a very important layer, one that is poorly understood, but which has lots of consequences with regard to our daily lives for any wood products that we use. Now what I'd like to do is to talk to you a little more about the cell types which make up the derivatives from the vascular cambium, and in particular, I want to focus on the secondary xylem. So for this discussion, we're going to look at a tree which is now making secondary xylem, and what I'd like you to do is going to be sort of dynamic. And we're going to start off with a cross-section of a tree, and here I'm going to put for a reference point, this is the center of the tree. The center of the tree is going to be located here. And on the x-axis, I'm going to put time. And we'll just have 4 different time points that we're going to put on here. We don't have to give them any names right now. Now let's talk about the vascular cambium first forming. This is a tree now which forms the vascular cambium the first time. So I'm going to draw the vascular cambium on this tree here at time, whatever we want to call this. The distance of the vascular cambium from the center of the tree at this point is x. Now we're going to wait for some more time to pass. If more time passes, the vascular cambium is going to have time to make cells, to make divisions. So here is our center again of the tree. And as I indicated before, as the vascular cambium now is beginning to divide, it's going to make some cells. So let's just say it makes a couple of large cells here. Here's the vascular cambium again. And so now the distance from the center is x plus y, because we've now added on these two cells from the outside. Now let's wait a little longer. We're going on in the season, we're going on in the year, and the cambium has had more time to operate. Here is the center again. Here is x. Here are those two cells which we drew before, which are y. And now we've made a few more cells, which we can say z. So the distance is now x plus y plus z toward the center. And finally, the last one, here's the vascular cambium again, in the last stage that we put over here, what we find is here is the center of the tree again. We have nothing here. Then we have our two big cells. Then we have two other cells. And now we've had some more time, and we notice that the cells are getting smaller. So this is x plus y plus z plus a. That's the distance from the center. Distance from the center that we have moved. Everybody with us at this point? Okay, this structure over here, which has been produced, is actually the structure that is formed in a tree over a period of 1 year. So for these marks over here, we can say that this is winter, spring, summer, and fall. And now what I'd like to do is to look at one of these years' worth of growth in a little more detail. So here is the center of the tree. Here's the vascular cambium. And if we look at this in a little more detail, we find the large cells that we had before, and then we find some smaller cells. So this is now 1 year's growth. What is this structure called? This structure, which represents 1 year's growth, you know of as an annual ring. An annual ring. So the cell types that are being produced over a year's period of time. And what I want to comment on now, and I want to draw your attention to, is I didn't draw all the same cell types for the entire length of the annual ring. This is all secondary xylem, and these are vessels. Now what I want you to notice on them is that the vessels that have been produced in the springtime are very large. And as we move into the summer and the fall, the vessels become smaller and smaller until when we reach winter again and the conditions outside are not favorable for growth, no new cells are produced. Now, why do the vessel sizes diminish in a typical annual ring? The reason they diminish is that in the springtime, which is the best time to grow, lots of water is available. And so the plant takes advantage of this by making very large vessels. As the summer approaches, the amount of water and the conditions begin to turn less favorable. So by the time you get into fall, where water is less available and temperatures are cooler, you are now developing fewer and fewer large elements of vessels. And this is a reflection of the differences in the climate for the year. Now, if we look carefully at a tree which has made several years' worth of growth, what we would find is the following. So what I'm going to do is make 3 years worth of growth. Here is the center of the stem again, the center of the stem. And what we would find in our particular tree is that we would have large, small, 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 smaller. And then we have large, small, 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 smaller. And the same pattern in a typical year would repeat. Large, large, small, 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 small. We have 3 annual rings here. The important point is that when you look at a tree in cross-section, what your eye picks up, the reason you can see an annual ring, is because you are seeing the gradation in size of the vessels, or the vessel elements. Now, in this particular tree, we have drawn a repeated pattern of large in the spring and small in the fall. What you should understand, it's important that you understand from this picture, is that the vascular cambium is on the outside. You should make sure you understand where the vascular cambium is. So we have 3 annual rings here reflecting 3 different periods of growth. For those of you who are interested in going into anthropology or into archaeology, tree rings are commonly used by anthropologists to study various cultures, especially cultures which have been subjected to very stressful conditions or cultures that have disappeared. Individuals who study tree rings are known as dendrochronologists. And by studying tree rings, that is the width of the ring, the number of large vessels or small vessels, one can tell something about the climate of a particular period of time. And this has been useful in understanding why some North American native populations have disappeared from areas, because disappearance can be traced back to beginnings of droughts. So this is an important resource for an anthropologist. Now finally, before I show you some slides of this, I want to make sure that you understand that not only are we making secondary xylem, but that we're also making secondary phloem, or bark. So let's begin with sort of a summary where we put our vascular cambium here. And let's pretend like this is going to be a 3-year-old tree. So we're going to put onto it 3 years' worth of growth. This is secondary xylem. Here is the center of the tree. 3 years' worth of growth. And now on the outside, we're going to put on secondary phloem, which is also made annually, but is more difficult to pick up. 1, 2, 3. So here we have 1, 2, 3, and here we have 1, 2, 3. This is secondary phloem. Now, the important point for you to understand, and this I'm asking you to go inside the tree to understand this, is that the oldest layer of secondary xylem is the deepest inside. This has been produced the longest time ago. Everybody clear on that? The oldest layer of secondary phloem is to the outside. They're mirror images of each other. The layers that are closest to the vascular cambium are the newest, the most recently formed layers. This is an important concept for you to understand, a difficult concept. Let's look now at a couple of pictures of this to maybe solidify it a bit more in your mind. Okay, this first slide shows you the cross-section of a stem showing primary growth. Here is the phloem and the xylem, and in this region here of an individual bundle, we're going to have the vascular cambium forming, and the vascular cambium is this layer of cells which is now running between the bundles, but forms between the phloem and the xylem first, starting to push the phloem and the xylem apart, so that in a later slide, what we see here is that the phloem and the xylem now are no longer connected to each other. The primary phloem is pushed all the way to the outside, and the primary xylem now is being pushed to the center of the stem where the pith is. Here we have now a much older tree showing a lot of secondary growth. These purple cells are secondary xylem, large vessels. Secondary phloem or bark is being produced here. We have the primary phloem, the cortex, and the epidermis. And here is a much older one where we have now the large layer of secondary xylem. The primary xylem would be toward the center here where the pith is located. And finally, I wanted to show you a picture of this tree, which you're going to actually see in the lab. This is a tilia tree or linden tree. This particular tree has 3 or 4 distinct annual rings. Here is the center of the tree, and if you count out, you can see there's 1 year's worth of growth, 2 years' worth of growth, 3 years' worth of growth, and in the 4th year, the tree was cut down. So this tree has 3.5 annual rings, and this is a slightly different tree, but of the same type. We're going from the center, the oldest ring 1, the next oldest 2, 3, and then it was cut down in its 4th year. And the location of the cambium would be where the secondary phloem and secondary xylem come together. Here is a tree now which has produced large vessels right at the beginning of the season, and then as the season has worn on, there's been drought or whatever, less water, and fewer large vessels are produced. You should understand that the vascular cambium is located on this side, not on this side, and you can tell that by where the annual rings end and begin. Here is a cross-section now of a gymnosperm. Gymnosperms, as we said, don't have vessels. They just have tracheas, but they still show secondary growth. And again, you can see annual rings because the first formed elements of the annual ring have thicker walls on them. Finally, I wanted to show you this slide, which has a picture of a cross-section of wood to indicate to you that there are different sizes of the rings depending upon what time of year the tree was cut down. And in this particular case, looks like 95, this early part, or 85, the late 80s were a pretty good year for rain. And then as we moved into the 90s, there was less rain until we moved up here to the late 90s, where there was considerably less rain, and it's reflected in a difference in the size of the annual rings. Okay, we're going to leave the screen down for a minute, but if you could turn the light on. Now, what I want to do is to discuss for you some consequences of the operation of the vascular cambium. Many of you are familiar with these consequences. For example, when you were a kid, if you pounded a nail into a tree, if that nail went into the tree deeply enough and you then moved away from your neighborhood or didn't go back to that particular region, assuming the nail didn't fall out, it would be embedded in the tree. The tree would have swallowed up the nail. For those of you who may tie a rope around a tree when you were a kid and then go away, if you come back many years later, that rope will have been broken. The gradual increase in girth of the vascular cambium puts pressure on that rope and eventually will break it. And I'll show you a picture of this in a moment. But perhaps the most interesting example of a consequence of the vascular cambium is shown by this structure here, which is a knot hole. Many of you have seen knot holes, not realizing where they came from or how they formed. But they are integral to understanding wood. If you go outside, as I hope you'll do after the lecture day, and look at trees, especially the redwood trees, what you will find on the redwood trees is that they have branches, but the branches don't seem to begin until about 10 or 15 feet above the ground. What happened to the branches which were lower down when that tree was smaller? So let's make a longitudinal section of a redwood. And on this redwood tree, we're going to put a branch, and we're going to say that this branch is dead. Maybe some animal chopped off its apical meristem. It's no longer going to elongate. Now let's let some time go past. And as this time goes past, here is the vascular cambium. The vascular cambium is now operating, and now the tree has gotten thicker, gotten wider, because the vascular cambium has been operating. Here is the vascular cambium here, and here's the vascular cambium here. What happened to that dead branch? That dead branch, essentially, was swallowed up into the tree. It was incorporated into the tree. And so when you make a cut in the lumber mill, and you make a piece of wood, a cut from the piece of wood that was where the dead branch was, that dead branch is going to be the knothole. It is dead tissue which has been surrounded by living tissue as the vascular cambium has continued to grow. Let's look now at a few pictures of these phenomena. Okay, this first picture shows you a tree around which we have tied a rope, and probably we could dim the lights a little, tied a rope, and when this rope is tied around the tree, the increased pressure from the tree will gradually make the rope break. Here is the broken rope as the tree has increased in girth as a result of cambial activity. Here is a beech tree right outside, and this beech tree had a branch cut off, and we're going to look at a high power of this tree. Here is the branch which is cut off, and the vascular cambium is now flowing over this particular cut. So if we look at it, and by the way, this is Rita and Mike. I expected something to happen to them, that they would move apart as the bark got bigger. But in fact, Rita and Mike are still together. And I'll show you, this is 10 years later. That particular hole now has been covered up as the vascular cambium has produced more wood and is swallowing it up. And if you make a comparison between the two, you can see now the way the hole is getting smaller and smaller. So in 2013, pretty soon that hole will be entirely covered. Here's an interesting one. Somebody must have taken their bicycle, put it over a branch, and then left it. And came back many years later, and the tree now has grown around, the cambium has grown around the bicycle and has become embedded in probably wood. Here are the branches of the redwood tree. These branches are dead. They're no longer going to elongate. And if you hold this piece of wood that I have up here in front up and imagine that this piece of wood is going to be coming from the inside as this particular tree swallows up the wood, this is the origin of the knothole. Finally, I want to just take you to the eucalyptus grove, which is right by LSA. I think all of you are familiar with it. When branches are cut off of the eucalyptus tree, they no longer have apical meristems. They're no longer going to be elongating. And as the tree continues to grow, then it's going to swallow it up. Here is a shadow of a branch which was cut off a long time ago. We're going to look at a little higher power here. Here you can see that branch, which has now been swallowed up by the vascular cambium, and eventually it will be totally obscured as the tree enlarges. Okay, let's now put the screen up for a few minutes. We can have the lights. So we've talked about the vascular cambium, and we've said that it is a tissue or a cell layer, which is what we think it is, which increases the diameter or girth of the tree. We don't understand how it operates, but we know that the massive dimensions of trees are due mainly to the activities of the vascular cambium. But there is another way in which trees can increase in girth in the eudicots and in the gymnosperms. So we're going to look at the eudicots and the gymnosperms. And we can get an increase in thickness due to vascular cambium activity. And we said the vascular cambium is a lateral meristem. It runs up and down the length of the tree, a lateral meristem, not an apical meristem. But there's also another way of increasing the diameter of the tree. And it is due to a second lateral meristem. A second lateral meristem which forms. This second lateral meristem produces material which not only increases the width of the tree, but also protects the tree. So let's look at a cross-section, let's say the eudicot tree. Here is the center. Here is the vascular cambium. Here would be secondary xylem. Here we would have secondary phloem, just as you've seen before. And depending upon the species, somewhere in the secondary phloem, this new lateral meristem forms. This new lateral meristem makes a material which protects the tree. All of you are familiar with this material. This new lateral meristem makes cork. Cork acts as an insulator, as a way of sealing the tree. It is impregnated with suberin, the material that we talked about before that prevents water movement, and it makes a very tight seal. So here we have cork. Makes a very tight seal around the entire tree. And because it's so protective, so difficult for anything to get across, this poses a problem for the tree. It is so strong and so protective that oxygen cannot enter into the tree. And yet, we have the vascular cambium deep in the tree, which is really dividing rapidly, and it needs to respire. It needs to get oxygen. How does oxygen get in? Well, oxygen gets in by the production of pores, which go through the cork. Pores which travel through the cork. And when the pore emerges on the outside, it makes a structure that you're going to see in the lab, which is called a lenticule. A lenticule. Now, air can go through these pores, but as it turns out, nothing else can go through the pores. No water, no nutrients. Nothing can move through the pores either direction other than gases. So what this means then is that the tissue outside the cork is all dead. Can't get any water. Most of the cork commercially in the world comes from this tree here, which is the cork oak. For those of you who've been to Davis, the Davis Quad is surrounded by cork oaks. We have some here on campus. Most of the cork comes from Portugal in the world. It's used in many products, linoleum, wine bottle corks, asphalt and freeways. A lot of uses for cork as an insulating material. It's almost indestructible. Now, what I want to do is to tell you a little bit about the corks that are used for winemaking. In Portugal, when the cork is harvested for winemaking, the original cork produced by the tree is a jumbled sort of mixture. And very skilled workmen see this cork, which is jumbled, cannot be used for cork for wine bottles. And so what's done is the cork is stripped off of the tree, and it's stripped off carefully so that the cork cambium, the cork meristem, is not destroyed. And in the process, then, we wound the tree, and now the tree is let to rest for about 10 years, and new cork reforms. And after about 10 years, this cork is stripped off of the tree, and then the tree is allowed to repair itself for another 10 years. And this goes on until the tree is about 130 to 150 years old. This is the way we get our cork. Now I have a question for you. Here is the cork from a wine bottle. Based upon the characteristics that I told you about the cork, I wanted you to tell me whether, if you were going to make cork for a wine bottle, whether you would drill the cork out of this way, like so, would you drill the cork this way, like so, or would you drill it this way, like so, tangentially? Which of these three ways, this way, or this way, would you take the cork from this particular structure in order to have the highest quality cork which would seal your wine? Think about that based on the characteristics of the cork. So these are the cork, and it is an important region of the plant. The cork is frequently found on plants which grow in Mediterranean climates like we have here in California, where there are fires that come through the grassy areas, and for a very short period of time, there's intense heat around the base of the tree. And during that period of time when there's intense heat, the cork then protects the plant from the fires and from the heat which comes from the fires. Let me now show you a few slides of the cork oak. This is taken from the campus at Davis, where you can see the cork growing on the tree. Here is a cross-section of the tree, and here is the layer of cork toward the outside that you can see it. Here is a lenticule. So here's a cross-section of a plant. This is a lenticule through which air can enter or CO2 can leave, but water and nutrients cannot travel through the lenticule. And the last slide here just shows you cork, which has been harvested from the cork oak, being loaded on to ships. Now finally, I'm going to leave the screen down. Finally, what I want to say is that we have said that eudicots and gymnosperms have secondary growth. Monocots, as we mentioned before, do not have secondary growth. So monocots lack secondary growth. And yet monocots can develop into very large trees. For those of you who see palm trees growing outside, palm trees can get really big dimension. How is it that a palm tree, which is a monocot, can get to be a big tree? Well, it is able to get to be a large tree not by developing a vascular cambium, but by taking on other sorts of processes. And these three processes are: to get to be a big tree, it enlarges pre-existing primary cells. So it just takes cells which have been formed from the apical meristem, makes them bigger, so that increases the dimension. The second, which we'll show you pictures of in a moment, is that when the leaves fall off, the petioles remain attached to the tree. And many of you have seen this, the beautiful patterns on palm trees where we have various crosses and various different sort of patterns. That's a result of the petioles staying attached to the tree. And the petioles remaining on the tree increase its apparent diameter and also gives it strength. And the third reason that monocots can get thicker is that they form, and I'll show you a picture of this, they form roots on the stem, as we already mentioned. They form roots on the stem, and these roots also act to increase the girth or the diameter of the stem. Let me now show you a few pictures of these. Here is a palm tree growing here in Berkeley, and what I want you to notice is that the stem is thick, but it's more or less the same diameter as you go up all the way to the top of the tree. If we look more closely at some of the palm trees, this one has had some of the leaf bases taken off. There at the bottom half of the tree, the leaf bases remain, and at the top half, the leaf bases have been removed. And notice the difference in the diameter of the stem when the leaf bases are attached or not. And finally, I want to show you this slide of a palm tree in which the cells, which have been pre-existing, have begun to enlarge, and these enlarged cells now increase the diameter of the stem. And in the last slide, I want to show you this of the palm tree, where we have the leaf bases here. The diameter of the stem is relatively thick, but if you look down here toward the base of the tree, it's a little bit thicker. And if we look at this in high power, the reason for the increase in thickness is because we have many, many roots that have formed on the stem. They coat the outside of the stem and thereby increase its thickness and its strength. Okay, we'll see you then on Wednesday.